Assalamu alaikum class. Today in our course of Fundamentals of Genetics, we will look at the part 2 of Mendelian's experiment. But before starting that, let's just look at the learning objectives of today's lecture. In this lecture, we will learn about the analytical approach that Mendel used during his experiments with pea plants. In this lecture, our learning objectives are to describe the structure of a gene and how offsprings inherit genes from each parent. We are also going to distinguish between the phenotypes and the genotypes of an organism. And then we will describe the Punnett squared approach to a monohybrid cross. So let's look at the part of our previous lecture on Mendel's experiment prepared by Dr. Sadaf.
Mendel's analytical approach. We have already gone through this slide, but once again I'll repeat it just to link our two lectures. There was a question that why one of the two traits they disappear in F1 generation, and the answer to this question is concluded after the observation of F2 generation. The recessive traits and its unit factor do not actually disappear in F1 generations. They are merely hidden or masked, only to reappear in 1/4 of F2 generation. I have already discussed this pattern that recessive traits they do not disappear in F1 generation. They are just hiding there and they have to express in F2 generation. I've already told you in this one slide here that this white color flower is a recessive trait. It is not hidden in F1 generation. It is masked actually. This character is then transmitted to F2 generation, and here it reappears as 1/4 of the plants. So this character is recessive. That's why it is just hidden and masked over there. So just going to our lecture, today's lecture. There is a problem. I'm going to discuss it with you, and this is a sort of your assignment, you can say. You have to solve it your own. You have to make Punnett square for each and every cross. Let us discuss it. These are the crosses given for pigeons, and they are existed in checkered or plain colors. Um, Checkered or plain colors patterns. These are the P1 crosses. Checkered, when mated with checkered, the F1 progeny gives 36 checkered and no plain pigeon, the resulting progeny. When checkered was mated with the plain pigeon, plain is the color pattern, okay? The resulting progeny was 38 checkered and no plain color. When plain was mated with plain, here you can see there was no checkered, all were plain. So based on these values, you can easily determine which character or a trait is dominant and which one is recessive. So when these F1 generations are mated together in different patterns, here you can see the results are when checkered A, the checkered A will be resulting here. This one will be the A. Okay? Here, this checkered A when mated with plain C, this one, plain C, the resulting F2 progeny is all checkered and no plain. In the second cross, checkered B, this is the checkered B results. F1 checkered B. Now you can see here, checkered B with plain C. The resulting progeny is 17 checkered and 14 plain. Here you can see almost half, right? So this is just to tell you that you have to go through this example and you have to solve it based on these values. You have to determine the trait, which one is dominant and which one is recessive. You have to calculate the ratios actually. So see this cross F in which they have taken checkered B, this one, and checkered with checkered B again. Means they were selved. No, not selved. They were crossed with one another, checkered B pigeons. The resulting progeny is amazing. 28 checkered, 9 plain. And the last cross is checkered A, this one, is mated with checkered B, this one. And here comes 39 checkered and there is no plain. So you have to make the crosses of all these combinations and you have to find out that how are the checkered and plain patterns are inherited. Select and assign the symbol for the genes involved and determine the genotype of the parents and offsprings in each cross. So I will tell you, like for checkered you can use C, C, and for plain you can use P, P, the two alleles for plain and CC for checkered. Okay. So another important thing is during his experiments was a test cross, and this is a very important point here. So test cross is basically a simple method that is still used today to discover the genotype of plants and animals. So the organism expressing the dominant phenotype but having an unknown genotype is crossed with a known homozygous recessive individual. Here you can see this is the flower, purple color flower, which is under consideration, or this is the test organism for which we are going to make test crosses against it. So normally we know that this purple color is a dominant phenotype, but we don't know the genotype. What is the allelic distribution? That was unknown, right? So we have to cross this plant with homozygous recessive, because we know if a plant is homozygous recessive white, it surely means that the genotype of this plant is small p, small p. This is understood. But for this plant, you don't know either it is capital P, homozygous or heterozygous. In both of the cases, it has shown the purple color. That's why we have to cross it with homozygous recessive white. So this process or this test cross is very analytical, and Mendel's analytical reasoning must be considered a true outstanding scientific achievement on the basis of rather simple but precisely executed breeding experiments. He not only proposed that discrete particulate units of hereditary which exist, but he also explained how they are transmitted from one generation to other generation. And apart from that, these test processes are basically explaining or explaining the reasons or explaining that how can we type the genome or allele distribution of the unknown allelic combinations of the plants. So when this purple color plant was crossed with homozygous recessive white, all of the four individuals here you can see if the result, because we don't know the genotype, right? That's why I'm saying if if all of the four individuals, or if F1 generation from here, all of the offsprings have purple color, when this purple color flower is crossed with the homozygous recessive, and the resulting progeny showed that half of the offsprings were white and half of the offsprings are purple. It means that somehow this combination small p has taken place in this one individual and in this one individual. For this plant must carry one small p allele. That's how we can easily conclude what will be the genotype of that plant. If the resulting progeny is showing this ratio 1:1, or you can say it 2:2, 2 purple and 2 white. In this case, you can identify the genotype of this flower plant, which is heterozygous dominant with capital P and small p. Similarly here, when there are all purple color flower plants, this is a very simple thing that this plant has produced two gametes which are having capital P, and you can easily denote the genotype of this plant, which is homozygous dominant, capital P, capital P. Now, here is an example. If we have two plants, tall and dwarf, both are the garden pea. We don't know that this tall is homozygous tall or heterozygous tall, but we surely know that this is homozygous short, right? So if we make a checkerboard, here we have both tall plants. Phenotype is observable. You can see them tall, right? This is tall too. But we don't know the genotype. We have to see what progeny they will give. Suppose if they are giving all tall plants, what does it mean? What will be the genotype of this plant? Capital T, capital T. That's why they have given all T, 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 T here, right? This is, we will write it here, the allele combination. This will be having again capital T, capital T. This. What happens if the combination or the pattern or the new offspring is not tall? If this plant is shorter one, then what will be the allelic combination? The allelic combination will be heterozygous tall. This one will be heterozygous tall and this one will be heterozygous tall. How? T, small t. T, small t. What will be the allelic combination here? Capital T, capital T, capital T, small t. Heterozygous but tall. Again, capital T from here, small t, and then this is the combination actually, small t and small t here. This is giving the ratio of 3:1 phenotypic ratio, whereas 1:2:1 genotypic ratio of this test cross. Again, we'll do it. If we have a plant that is tall, but we don't know what is the genotype of this plant, and we have a short plant. For this plant, we know that the genotype of this short plant must be small t for the character of height. So the resulting plants are half of the plants are tall, half of the plants are short. It means that this plant must have TT and this plant must have TT, small t, small t. So you can easily write small t here, obviously small t here because this is having two small t's. And now you can easily solve the problem with putting here capital T and you will get the allelic combination of this plant, which is heterozygous tall. Heterozygous tall, right? And both of these two tall plants are actually heterozygous tall plants. This is their genotype. Now, there is a fourth and last postulate of Mendel's. Before that, we have covered three postulates of Mendel's law of experiments. And this is the independent assortment, in which uh, the law of independent assortment says that the allele pairs separate during the formation of gametes. This means that the traits are transmitted to offsprings independently of one another. In other words, when two independent events occur simultaneously, the probability of the two outcomes occur occurring in combination is equal to the product of their individual probabilities of occurrence. So, in the last figure, as I have told you, these are the genes, and before the meiotic division, the homologous chromosome has to duplicate and pair up. I may show you the previous slide where we have this one. Okay, so they have paired up here, right? Similarly, this capital G, and small g, they have duplicated. Like I have already told you that paternal and maternal chromosomes, they have to duplicate during first meiotic prophase. And now the same chromosomes or the sister chromatids are going to now are going to detach from here and have to move apart for their independent assortment. Here you can see this G is going is now one of the gamete, and this is the combination of capital G with the capital W, small g with small w, capital G with small w, and small g with capital W. So these are the different combinations, because in meiosis a single egg cell, it divides by meiotic two meiotic divisions, and then it produces four gametes. This is how the four gametes are equally different from one another genetically, right? So all possible genetic combinations are found with equal probability. And now this gamete has a different chance of getting fused with the another partner's gamete. So now we see Mendel's dihybrid process, which generate a unique F2 ratio, and this ratio is very different from the monohybrid cross. As a natural extension of monohybrid cross, Mendel's also designed experiments in which he examined two characters simultaneously. Similarly, we can have trihybrid crosses. Such a cross involving two pairs of contrasting traits is dihybrid cross or two-factor cross. Dihybrid cross can have two contrasting traits. For example, here you can see in this P1 cross, for the color of the seed and the shape of the seed. Color and shape, these are the two contrasting traits. And in this cross, they have taken yellow as a seed color and round, which is the dominant character, yellow and round, both of the characters together, and then green and wrinkled. Both of these characters are actually recessive characters. So P1 means parental generation 1, and this cross is showing the cross between truly breeding yellow round plant, which is producing yellow colored and round seeds, and truly breeding yellow round with truly breeding green wrinkled. So let us see how this cross is made. Okay, so here you see this is the P1 generation and represented by capital R for the round shape, capital Y and capital Y for the yellow color. This parental generation is small r for wrinkled shape and small y for the green color. Truly breeding means all four alleles are similar. Homozygous for round and yellow, homozygous for wrinkled and green. So in the F1 generation, when there is a combination formed, they mix up together their alleles and they form capital R with small r, capital Y with small y. So it is again random. So this F1 generation or F1 plants were yellow and round. The combination is, for example, here I may write capital R, Y, Y, small r, and small y. There is equal chance of this R to go with this R to go with this for a single new offspring, this Y with this Y with this, or vice versa. In either case, if you take two alleles, capital R, small r, because this is the similar one, then capital Y and small y, the similar one, this one is the similar one. So with this combination, the F1 plant will be capital R, capital Y, small y. Okay, so here is the Punnett square. This is one of the F1 generation plant and this is one of the F1 generation plant. As I have made these gametes, just if you put capital R, capital Y, small y, and cross it with the similar parent, you can get different gametes. So you can have a combination of capital R, capital Y, capital R, small r with the capital Y, and small r with the small y. Similarly, this individual will be having all these similar kind of combinations of their gametes like this one. Okay, so here you can see what will be the combination here. This is round and yellow and is homozygous. This is round, homozygous round, but heterozygous yellow, right? So this is heterozygous round, but homozygous yellow. Again, this is heterozygous round and yellow. Keep it in mind. This is heterozygous round and yellow. This one, this one, and this one, and this one. All these are heterozygous round and yellow. We have to count more heterozygous round and yellow. These are the four one. Okay. So heterozygous round and yellow. These were again yellow and round, yellow round, yellow round. So all these yellow round are 1, 2, 3, 4, 5, 6, 7, 8, 9. This one. So just see here. This is homozygous round but green. Homozygous round but green. Homozygous round but green. This one. Here you can see the combination wrinkled but yellow, wrinkled but yellow, wrinkled but yellow. This one. And the last is wrinkled and green. So this was actually the phenotypic ratio. The phenotypic ratio, which is different from the monohybrid cross, and this is 9 is to 3 is to 3 is to 1. 9 yellow round is to 3, green round is to 3, yellow wrinkled is to 1, green wrinkled. So in that dihybrid cross, we have to now calculate the genotype of that hybrid cross. I have to erase these lines first. Okay. So for genotype, let us see what is its genotype. Here we will see. Genotype will be 1 out of 9, out of these 9, 1 was homozygous round and yellow. These two were homozygous round but heterozygous yellow. 2, right? This one and this one is heterozygous round but homozygous yellow. Again, 2 of similar genotype. And then this is these 4. I have to draw a line once again. These 4 are heterozygous round and heterozygous yellow. All these 4. So the ratio is 4. And then out of these 3, you can see one is homozygous round and homozygous green. Homozygous round and homozygous green. Again, 1. And then 2 of these round green are heterozygous round, homozygous green. So we have 2 individuals of the same genotype. Among these 3, you can see this is homozygous, wrinkled and yellow, right? So again we have 1, ratio 1. And 2 are homozygous, wrinkled but heterozygous yellow. These 2. Here again 2 individuals with the same genotype, and then only 1 with homozygous, wrinkled and green. 1. So This is the genotype of these all 16 individuals. 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 and 4. So this up to here belongs to these 9. This belongs to this one, and this genotype belongs to this phenotype. So when you calculate it, 4, 6, 7, 8, 9, 2 and 1, 3, 2 and 1, 3 and 1. You can easily calculate it by cross-checking this genotype with the phenotype. This ratio 9 is to 3 is to 3 is to 1 is actually the dihybrid ratio, whereas the previous one 3 is to 1, which was used for monohybrid crosses, is known as monohybrid ratio. So you have to remember this one, that for dihybrid crosses the ratio is dihybrid ratio, and for monohybrid crosses the ratio is monohybrid ratio.
So, after watching this lecture on the analytical approach that Mendel used while performing his experiments with the bee plants, the main key takeaway points are: first of all, genes are passed from the parents to the offsprings. Mendel used bee plants with seven distinct traits or phenotypes to determine the pattern of inheritance and the underlying genotypes. Mendel found that crossing two purebred pea plants which expressed different traits resulted in F1 generation, where all the pea plants expressed the same trait or phenotype, like the seed color or the seed shape or the color of the flowers. When Mendel allowed the F1 plant to self-fertilize, the F2 generation showed two different phenotypes, indicating that F1 plants had a different genotype. Similarly, fertilization between two true breeding or purebred parents that differ in only one character is called a monohybrid cross. For a monohybrid cross with the two true breeding parents, each parent contributes one of the two types of alleles, resulting in all of the offspring with the same genotype. Dihybrid cross is a cross between two true breeding parents that differ in two observed traits. While a test cross is a way to determine whether an organism that expressed a dominant trait was a heterozygote or a homozygote.